So Zhang Ziqiang from Taida Senisu. Today I'm gonna share with you how to manipulate sphingolipid enzymes and see the sphingolipids, which is still a very challenging task in vivo. Okay. So brief keywords about my lab. We do neurobiology of aging and disease. I'm a Fry geneticist. I'm interested in cell biology and specifically intracellular trafficking. My recent focus is on sphingolipid. And besides those cell biological studies, I also collaborate with people from the clinical side, including studies on neurodegenerative diseases such as Parkinson's in National Taiwan University Hospital and some rare disease in the very good hospital, Veteran General Hospital. And so today I'm going to talk about sphingolipids. This is the only beautiful chemical structure that I'm going to show because that's the only structure that I know. So I took general biology twice in college. So and what do I know? The guts that I probably am not good enough. So it took me here to study it like forever. And sphingolipids are use sphingosine as a backbone. You add fatty acid and you make ceramide, and you add phosphorylcholine or phosphoethanolamine, and you get sphingomyelin. And you add more sugar. That's my understanding about chemistry. You add more sugar, you get those complex sugar-like sphingolipid for the cerebroside, etc. So if you look at those degradation and the formation of those complex things, and they just form like Taipei Jieyun, like every step does something like to change to convert one thing to another, and the central block is the ceramide that's gonna make sphingomyelin. That's important for neuroscience. That's all I know about those network. So I do know that a lot of mutations happen in almost every step of this network, and those mutations are important because people sick because of those mutations that like ALS, epilepsy, and Parkinson's. Those are big words that people really know. And my previous focus was here in the middle. That's about like 7 years of work. We focus on this enzyme that makes ceramide, and we study the cell biology because it's related to a disease called leukodystrophy. And we use Fry genetics and combined with confocal imaging and molecular biology to understand it, and that's our past work. And so focusing on one disease and one gene that can get us to beautiful mechanisms, but now from that part, we suddenly one day we just step back and look at the whole network and say that, hey, Fry and human are quite similar, and those networks are conserved between Fry and human, and why don't we just study them all? And that's one crazy student that be crazy. So I just so there's a word called you jump, I jump. So he wanna jump, so I had to jump. So that's in human, that's 100 something enzymes. In Fry, it's a little bit less. It's about 53. So we work on 53 enzymes. So we're gonna study 53 enzymes in a complex tissue called the brain, because the brain is the most heterogeneous tissue in our body. It's heterogeneous in terms of spatial organization, like different parts, different compartments, and also developmental dynamics. It changes a lot. So I wanna do 50 genes in the brain. Okay, so how are we gonna systemically study 53 genes? So that's our strategy. So first we modify those 53 locus by knocking a fragment in the endogenous loci that's gonna make a tagged protein as well as a transcriptional activator called Gilmore. It's a right term that's gonna activate something. Okay, so that allow us to do systemic gene profiling of the whole network. And we also wanna do functional analysis by manipulating with those enzymes by either overexpression or knockout. And for knockout, we wanna do targeted protein expression. So for the second part, I'll talk about the functional analysis by introducing you with a degradation system that we just made. And also we wanna see the lipid. And so we made a probe that's gonna recognize sphingomyelin, and I'm gonna show you how that works, okay? So the first part is to build a 4D map of the metabolic network by visualizing those enzymes. Visualizing enzymes are not hard, it's just a lot of work. So, so those are 53 brands, and as you can see, that if you squint a little bit, you see that not every single cell expresses everything. The heterogeneity is there. We know this from textbook, but I'm showing you here. Okay, we saw it, the heterogeneous expression, and from those heterogeneous expression, we can deduce some rules. Like so, this is a mRNA level, and this is protein expression. Protein and mRNA mostly are expressed in the same cell, but in some cases, in some gene, they just express differently. So that would suggest transportation or differential degradation. And those are some rules that we are interested in. And so what we have done here is to profile the CNS and also PNS, the peripheral nervous system in the adult. And also we are challenging the flies with those high sugar. It's quite interesting, making them eat more sugar. It's just like my kid likes Coke and they just drink too much. So I want to tell them it's not good. So I want to do some experiment to show them, hey, it's bad. Okay, so and also ongoing thing is we are doing Parkinson's by introducing Parkinson transgenes and also Alzheimer's transgenes. And we have collaborators that's interested in the reproductive system and also neuromuscular junction, all are bright people. And we are happy to share the platform. That's the first part. Second part is to degrade the protein. So the degradation, the concept is easy. You just fuse a nanobody to an E3 ligase, and then those two would specifically recognize the protein and then for degradation. And this, since we have knocked in our protein with HA10, we can use an anti-HA nanobody, fuse an anti-HA nanobody to E3 ligase, and then we call it degrade HA, and then we do those degradation in vivo. Okay. The in vitro or cell culture degradation has been shown previously, but we do this in a living organism. And here we show that this protein expressed here, okay, in 2 minutes, and the degradation works quite well. The third part is to visualize the lipid. And in the interest of time, I'm just gonna say that this probe recognizes sphingomyelin. We always express the gene that produces sphingomyelin. The probe is getting greener. We knock out the gene and the probe, the signal is gone. And the probe recognizes the lipid raft because it co-localized with the lipid raft marker, and the Fred experiment showed the energy transfer between the probe and the lipid raft marker. Okay, so our profiling told us there's probably not a single omnipotent neuron that does everything. In the textbook, we always see a cell that have all the network, everything is thrown on it. In real life or in your brain, in every single neuron, that's probably not the case. Okay, and how do cells communicate to transport the lipid or the enzyme to orchestrate the network requires further investigation, and that's where we are to understand the human brain in the tiny fry brain. So this is the whole network. I have 2 slides to conclude everything that our strategy and advantage of studying sphingolipid network in a living organism is that the network is highly disease relevant, and there are no mutations, and there are a lot of novel mutations nowadays. A lot of next generation sequencing has identified a lot of mutations, and we have identified genes of specific expression pattern. Those are reporter flags and those are lipid markers, and we are knocking out the entire network So besides the reporter flight, we are also making those CRISPR engineered those knockout flies. As you know that CRISPR, we do a lot of CRISPR work. And our strategy is to establish humanized fly model and then seek to elucidate the molecular mechanism. Potential collaborations, we would like to work with scientists with interest in sphingolipid, and we are building in vivo biosensors of sphingolipid. I'm just showing you one case, but if you have more methods to make more probes, please let me know. And people with expertise in lipidomics, bioinformatics, or you have novel mutations in sphingolipid enzymes, let me know, please. And with that, I thank people in my lab where the money is from and my collaborators. Thank you very much.